When we had our chemistry unit, you learned that nucleic acids were a type of organic compound, which means that they contain atoms of carbon. Nucleic acids are huge polymers made of smaller connected monomers called nucleotides. There are two types of nucleic acid, deoxyribonucleic acid, or DNA, ribonucleic acid, or RNA. For the test, you need to know the differences between RNA and DNA. First, the structure of RNA is called a single helix. The structure of DNA is called a double helix. DNA is a large molecule that cannot leave the nucleus. RNA are smaller molecules, three different kinds. They're formed by DNA, and they can exit the nucleus through the nuclear pores and are intimately involved in protein synthesis. So RNA is a single helix, DNA is a double helix. Next, both of these nucleic acids have 5-carbon sugar, but the 5-carbon sugar in RNA is called ribose. 5-carbon sugar in DNA is called deoxyribose. Then there are 4 bases that are found in RNA and DNA. Notice that adenine, guanine, and cytosine are found in both, but the 4th base in DNA is thymine. Instead of thymine, RNA substitutes uracil. Notice that I've capitalized the first letter in the name of the bases. Now the nucleotides all have a phosphate, a 5-carbon sugar, and some kind of base. The base is different. So we use the first letter in the name of the base to identify that type of base in the nucleotide, and we just call the nucleotide by A, G, C, or T in DNA, or A, G, C, and U in RNA. So you need to know this chart for the test. This diagram shows you the single helix that forms the structure of RNA and the double helix that forms the structure of DNA, and that cytosine, guanine, and adenine are the same, but the different base in the two nucleic acids are uracil in RNA and thymine in DNA. Now, as far as the historical development of the discoveries that were made along the way to determine the structure of DNA, you need to know every scientist that's in this section. To better learn these scientists and what their contribution was, go to your concept reviews in the DNA part of this module, and there is a concept review that allows you to match the scientists with their contribution, and you need to know those scientists and their specific contribution for the test. So I'm going to quickly pass down these scientists and come to Wilkins and Franklin. Franklin and Wilkins worked out of the same lab. Franklin did a lot of X-ray technology and did these X-ray diffraction pictures of DNA. That picture right here was shared by Wilkins without Franklin's knowledge to Watson and Crick and helped them finally develop their model of DNA. Now in 1953, Watson and Crick published their paper with the structure of DNA in it, and they had combined all of this research from these other scientists, put it all together, and developed their model of DNA. These two YouTubes give you a very good, quick summary of what Watson and Crick did and the contribution that Rosalind Franklin made to their discoveries. Now, the problem that Watson and Crick had when they built their model was to orient it correctly. First, they tried 2 5-3 strands, but they found that they wouldn't bond correctly. So they decided that it needed to have a 5'3" strand and a 3'5" strand, opposite polarity. So they had used, Crick had used this diagram, or X-ray diffraction picture of DNA that Franklin had taken, and he had determined the width of the DNA molecule had to be only 2 nanometers. So he put two purines together, again oriented in the 5'3" and the 3'5" orientation. But when he did that, the molecule was too wide. So they were using paper models, just kind of like this, and they put two pyrimidines together. Then the molecule was not wide enough, it was too narrow. Now Chargaff had determined that G always equaled C and A always equaled T. So Watson and Crick concluded that that was the only possible nucleotide bonding in the DNA molecule. So they put G with C and A with T, and guess what? They came up with exactly 2.0 nanometers for the width of the molecule. So they published their paper in 1953, won the Nobel Prize along with Wilkins in 1962, but Rosalind Franklin by this time had died of cancer, and the Nobel Prize is never awarded posthumously to anyone. Now these next two individuals, James Watson of Watson and Crick, headed up the project to map the human chromosomes and to determine the location of 20,000 genes in human beings, where they're located on the 46 chromosomes. It was finally decoded, and Craig Venter and Francis Collins, you need to associate their names with Project Genome, and announced jointly in 2000 that the human DNA had been sequenced.